Hello and welcome to another episode of the Omicscast. Today we're going to talk about synthetic genomics. We're going to review a blog post by Tom Ellis that gives a perspective of where synthetic genomics is today. Tom Ellis is a reader in synthetic biology at Imperial College London and ICL, and he's got a PhD in pharmacology from the University of Cambridge and an MSc and BSc at the University of Oxford before that. So he's got the Oxbridge duo, and then his experience along the way, most recently lectured in synthetic biology in Imperial College London, as you can read here. So in his blog post, he basically describes this assay in April 2019 for the Biochemical Society. He talks about synthetic genomics and highlights the pioneering work of George Church and Craig Venter, two big punchers in the wall of omics. Craig Venter, controversial in many ways. George Church, the biggest beard in omics. And he says, is it just synthetic biology with bigger bits of DNA? He says, no, well, no, not quite. And then he goes on and describes what he thinks synthetic genomics is in relationship with synthetic biology. Synthetic biology, he says, focuses on rewriting and reprogramming the DNA in cells. And synthetic genomics, he says, ways to produce new understanding of DNA and biology, either directly or by enabling new experiments that cannot be done any other way. How fast has this field advanced? Well, quite fast, or quite rapidly, I would say, in the last 10, 15, maybe 20 years, if you look at the values in this plot. Although there were some developments in the 70s, 80s, and 90s as well. And so Ellis describes mostly what has happened in the turn of the millennium and what it means and what could happen in the next 20, 30 years in the future. He says, Richard Feynman famously said in a quote, what I cannot create, I do not understand. And then he says, if you apply this logic to synthetic biology, then building up a synthetic gene system, a system of genes, would allow us to understand and mathematically model how these genes, how these genetic components interact with each other and create a cell, an organism, a working cell. And so he says, we already know and gone through the minimal requires for cells to genetically encode memory and rhythms. And when we discovered that, we made a first significant achievement in that part of science. And so then we're going to describe, following this block, a little bit of what has happened in which order. He says it's becoming routine to see dozens of genes used in synthetic DNA constructs. However, synthetic genome built from first principles from a kit of modular parts is still a long way off. And he says maybe a couple of decades. So in this plot here, human genome, or let's say a genome 10 to 20 times smaller flat genome, is still quite a way away. Modifying aspects of these, and we'll show examples of the Baker's yeast genome, we're there now. But a whole genome built from first principles, from a kit of modular parts, maybe another couple of decades away. We cannot yet write a genome from scratch, and so a lot of this is going to be editing and modifying parts of existing genomes and replacing the nucleus of a cell with a slightly modified version of the genetic components. And so a lot of this is going to help us in genome science, it's already doing, and in understanding new ways in which gene regulation, genomic regulation happens, which some of them are going to be unexpected, and they are already unexpected in many ways. And I had a video recently about TADs and CTCF and isolation marks A lot of what's happening in regulatory genomics now is in trying to find what regulates genomes and how genomes regulate themselves. And so it says, constructing, testing synthesized genomes and chromosomes that are increasingly different compared to natural genomes will allow us to understand all this genome biology, right? So one first aim of synthetic genomics is to deliver new knowledge of genome coding, genome content, genome organization. If we move things around in a genome, how does that affect the regulation of the genes among themselves, etc.? A decade of synthetic genome progress is that in 2008, there was a half a megabase mycoplasma genome constructed from chemically synthesized DNA. And then 2 years later, another mycoplasma, this time a 1 megabase mycoplasma that could replace a natural genome. All this work done at the J.K. Benter Institute. And there weren't measured changes to the DNA sequence. It was simply a synthesis and construction exercise, and they showed that it was possible, right? So synthesized genome, and then we're going to talk about other aspects of these recoding genomes, minimizing genomes, reorganizing genomes, and the modular genome and how to scramble things, etc. And so these are different papers, different achievements of Mycoplasma, E. coli, etc., things that have happened recently in the last couple of decades. In 2013, a team of Yale and Harvard showed a bacterial genome could be recoded, and by recoding here they mean removing all occurrences of rare codons used in protein synthesis in ecology. And so in the same way as we are used in popular culture to genetically modified organisms, GMOs, they coined the term genomically recoded organism, or GRO, for this genome where the information is roughly the same, but it's been recoded in a different way, right? And there'll be an analogy with this in computing terms later on that I hope you will find as familiar as I did. And so they said they altered these bases on a genome relatively large, 4.6 million base pairs. And so this wasn't a 4.6 million base pair synthesis exercise. It was a recoding exercise compared to the previous ones, the Mycoplasma ones. And they showed genomes could be made with recoding through their genes. E. coli and Salmonella bacteria, substantially more DNA recoding, constructed recoded genes from synthesized RNA rather than mutation. So this is more recent efforts in which you combine both. You combine recoding and synthesis rather than mutation, right? So you can have a large genome, change a few bases in it to recode it in one specific way here, change the rarest codons, or you can do recoding and synthesis all in one go rather than by mutation. Next step, genome minimization. 2016, again, J.K. Benter Institute constructed a synthetic redesigned version of the Mycoplasma genome before, but they minimized it. They took out the parts that were necessary. They left out genes and DNA that they deemed not to be essential for growing these in the lab in a Petri dish, which amounted to about half the genome, right? So this is like saying, let's chop out the bits that are not necessary and construct the minimal genome that is still viable, right? And beyond recoding, genome minimization. This is our most synthetic genome to date, according to Ellis, and then they also made a version where they the order and the layout of the remaining genes of the bacterial genome was totally changed. And here is where we are going to apply the same to the computing analogy, same concept to the computing analogy. What they did here essentially is made a version of the same genome where the order and layout of the remaining genes were changed in order, basically. And they changed them in, if you want, subjective manner. They arranged them according to their function. So they said, we're going to put, if this was in colors, all the blue genes in one part, all the red genes in another, all the green genes in another, etc. The order or the color of the genes here was according to function. All the genes that are related to this go in one bucket, all the genes that are related to performing this other function go into another. And then this team called this version a defragmented version of the genome, which is analogy to the process when computer files in a hard drive are relocated to common clusters to improve storage efficiency. And for those of you old enough, you will be familiar with screenshots like this, which is the defragmenting process of hard drive. As you can see, essentially the files in your hard drive can take different sizes, right? It's like having different boxes of different sizes in a room. The space in between the boxes is wasted space because a box that's 4 times the size of another one, if there's only 3 of the smaller ones, then there's a space in between that cannot be filled. So defragmenting is going from something that looks like this into something that looks like this, where all the yellows are nicely packed, and then all the grayish area is free to be used for something else. So in a conceptual manner, what they did is what in computers called defragmenting the genome. What about beyond bacteria? So we're talking about E. coli, Mycoplasma, etc. Where else can we go beyond that? Well, the synthetic yeast genome project called SC 2.0 created an 11 million base pair genome naturally split in 16 different chromosomes. So now it's not only one molecule, it's 16 of them. They did minimization, removal of unneeded non-coding elements such as transposons and introns. And it also had an element of defragmenting in the sense that all the tRNA genes were removed or are being removed from their locations of the main chromosomes and placed into a new synthetic tRNA chromosome. So this is like saying, we're not going to put all that we have in one basket. We're going to create a special pocket where we're going to put the tRNAs in there, right? A new synthetic tRNA chromosome. Furthermore, something that is quite interesting, I find it quite interesting, is this inbuilt scramble mechanism, where genes within the synthetic chromosomes can be randomly removed and rearranged given a chemical stimulus, right? So this is, the word scramble is quite a good term to coin here, because this is basically giving a sign to the synthetic genome to say, right, you can now scramble and rearrange yourselves randomly because I've given this chemical stimulus. This is a powerful method to exploit what genes are essential, if you think about it, because it means we can do random selection and random high throughput experiments to see what happens where the genome is reorganized in a specific manner, right? And what they found out is that the yeast genome can handle some serious rearrangement of its genes without many problems. I used to have a professor of microbial genetics in my undergrad that used to say microbes, or more specifically, most bacteria, are essentially a bag of genes. And they're a collection of genes put in a bag, and it doesn't matter what order they are, as long as all of them can be transcribed at some point, right? And humans, for example, are not like that. If you move a bit of the genome into another part of the genome, all sorts of wrong things happen, right? So the location of genes, the spacing between genes, the regulatory regions outside genes are very important to maintain function. And so yeast is somewhere in between bacteria and us humans. And with this scramble mechanism, they were able to find out more about how much of this random rearrangement of the genes was able to happen without causing problems to the survival of the rearranged genome. And so Ellis says there's clearly significant plasticity in the chromosomal structure and gene layout in a yeast genome. And so what's the next step here? We've done some bacteria, we've done some yeast, we've done some synthesis, some minimization, some defragmenting. We got tools to scramble the order of the genetic components, etc. What else can we go? Human and mice. So human cells grow in a petri dish, and mice, because they're a model organism very close to humans that we can do a lot of biology with. We want to understand what's the organization of regulatory regions, essentially. And we want to be able to do that and determine the gene function and how mutations lead to pathogenicity, right? And so all the tools that we develop in synthetic genomics that give us this power of associating genotype to phenotypes are going to be very important for human disease, things like cancer or cardiovascular disease, inherited hereditary diseases, rare diseases, etc. And so there says that it is important that there's a concerted effort or efforts to engage widely, disclose, and coordinate globally how synthetic genomics will advance over the next decades. And he mentioned this consortium, the GP Right consortium, to be able to do this in a coordinated fashion. Now, what kind of experiments are people doing today in this high throughput manner? Well, some of them are related to technology that's become very powerful, CRISPR technology, which allows to edit specific parts of the genome with a lot of precision, right? And if we design the experiments correctly, we can do it as a very high throughput method. And for those of you that heard about my other episodes in the podcast, you know that I talk about high throughput genomics, high throughput omics, sequencing, reading DNA. Well, this is high throughput writing of DNA and then doing some experiments afterwards. After which you go back and you just do another high throughput reading experiment, as it says here, read out by next generation sequencing to see what your experiments have rendered, right? And so the high throughput genome editing screening assays are basically trying to modify the genome at high throughput scale, test the outcome, and then from there infer the function of what has been modified. If we give a twist to this and we try and add it a component of drug discovery for big pharma and not so big pharma, we now have the opportunity to do high throughput genome editing with drug screening assays on top of it, right? So if we want to modify the genome, test the outcome, and infer the function, we can also modify the genome of a model organism, test the outcome, screen it with drugs, because the model organism is not going to complain about that, and then shortlist the therapeutic candidates. And I should say, this, like the previous one, the only difference that this previous one, it's a bunch of cell lines growing in a petri dish, and here we use a model organism. And I've chosen this from a new biotech called Gennomics, which uses zebrafish. There may be Germans involved, that's why it's also called Gennomics, in the sense that here we replace the cell lines with a model organism, the little zebrafish fishes. And so if we want to understand human diseases, we can generate zebrafish disease models, validate their phenotype, and then these pathological phenotypes treat with some drug, right? Some molecule library in a high throughput manner. And then we can do therapeutic candidate identification. Which of these many molecules are going to test this disease with gives us a good result. And we, evaluate the results of applying this drug. And so once we know this, then we can go to the equivalent gene in humans and say, well, there's an opportunity here to apply to humans and test it on cell lines and do the preclinical development. And so all of this is a write and read operation. Basically, we write genome by, in this case, custom model CRISPR modification. We do phenotyping. We do phenotyping in conjunction with drug treatment, and then we get a result. And this result we obtain by reading by next-generation sequencing what the result is. So as a conclusion to all this, a reminder of Feynman's learn by building approach, which is let's see if we can understand genome regulation by basically building genomes and seeing how the genes interact with each other. And that we can do high throughput genome editing and couple it with drug screening, combining high throughput genomics, the reading, and phenomics from the phenotypes that we've generated in writing the genomes. Thank you very much for listening. You can follow me on Twitter, twitter.com/Alberto Vilella. And this is also the link to my LinkedIn. You can also search for me, Alberto Vilella, on LinkedIn. I'm happy to answer any questions you have. And a reminder that this slide deck is available upon request. Until the next episode, goodbye.